Welcome to developmental biology. Imagine you are tasked with building a magnificent, towering Lego castle. Do you expect to open the box and find a microscopic, fully built castle that simply inflates when you add water? Or do you expect to find a single starting block and a complex set of instructions? This exact question puzzled the greatest scientific minds for centuries, and exploring it is the foundation of everything we know about life today. To understand how we grow, we must step back in time. Before we had powerful microscopes and an understanding of DNA, biologists had to guess how a baby formed inside a mother. This was not a trivial question. It was one of the most profound mysteries of the natural world. This led to a massive historical debate, split into two major camps, with brilliant minds arguing passionately for centuries. Let us explore the first of these theories to see how early scientists attempted to explain the miracle of life. The first camp believed in a concept called preformationism. This was the idea that all organisms develop from miniature, fully formed versions of themselves that already exist inside the sperm or the egg. It sounds like science fiction to us now, but some scientists even claimed they could look through early, blurry microscopes and see a tiny human, called a homunculus, curled up inside a sperm cell. They thought development was just a simple process of inflating this tiny body, much like blowing up a balloon, with no new structures being built. On the other side of the debate was epigenesis, a theory originally championed by the ancient philosopher Aristotle and later proven by scientists like Caspar Friedrich Wolff. Epigenesis is the scientifically correct concept that an organism develops step by step from a formless, simple mass, which we now know is the single-celled zygote. Instead of just growing larger from a prepackaged form, the embryo undergoes progressive differentiation. This means its cells divide, multiply, and take on highly specialized jobs over time to construct a complex, multicellular organism from scratch. Let us look at this diagram to truly visualize the difference. On the left, you see the preformationist view, a tiny human, or homunculus, packed inside a sperm cell, which simply inflates into an adult. On the right, we see the reality of epigenesis. Notice how it starts with a single zygote, which then divides into 2 cells, then 4, eventually forming a complex blastula, an embryo. It is a dynamic process of construction, not just inflation. This visual perfectly captures the shift in our biological understanding. You might be wondering why we even care about an old, incorrect theory today. Understanding this history is absolutely crucial because it highlights the shift from philosophical guessing to rigorous, evidence-based science. If we still believed in preformationism, we would assume every cell was permanently locked into its final form, meaning we would never have discovered stem cells. The triumph of epigenesis forced scientists to ask where the building instructions were hidden, leading directly to the discovery of DNA, genetics, and modern regenerative medicine. Now that we understand the biological importance of epigenesis, let us look at how it actually happens on a cellular level. Because the embryo is not preformed, it must build itself through rapid, successive rounds of cell division, a process known as cleavage. We can model this explosive growth mathematically to understand how quickly immense complexity arises from a single starting point. By applying mathematical principles to biology, we can track the exact number of cells present at any given stage. This brings us to a very important mathematical relationship. To model this explosive growth, we use a specific mathematical relationship. This formula perfectly illustrates the step-by-step building process of epigenesis. N equals N0 times 2 to the power of N. Let us break down what these symbols mean physically. N represents the total number of cells present in the embryo at a specific time. N0 represents the initial number of cells we start with, which is almost always 1, because development begins with a single fertilized egg. The variable N represents the number of division cycles that have occurred. Because each cell splits into 2 during a division, the base is 2. As N increases, the total cell count grows exponentially. Before we move on, let us see this with a concrete example. Suppose a frog has just laid a fertilized egg in a pond. We want to find out exactly how many cells will make up this embryo after 10 complete cycles of cell division. We know our starting point is a single cell, so N0 is 1. The number of divisions, N, is 10. By plugging these into our equation, we calculate 2 to the 10th power. The single cell has multiplied into a massive cluster, giving us a final answer of 1,024 individual cells. This actively demonstrates epigenesis in action. Now that we have used this formula to find the total cells, let us try working backward to prove how many divisions are required to reach a specific developmental stage. Imagine a sea urchin reaches the late blastula stage when its embryo contains exactly 4,096 cells. We know the final number, but we do not know how many division cycles occurred. We set up our equation by plugging in 4,096 for N and 1 for N0. This translates our biological observation into a solvable mathematical puzzle. Let us complete this mathematical proof to find the exact number of divisions. We have the equation 4,096 equals 2 to the power of N. To solve for the exponent N, we need to express 4,096 as a power of 2. By recognizing that 2 multiplied by itself 12 times equals 4,096, we rewrite the left side of the equation. Since the bases on both sides are now the same, their exponents must also be equal. We have mathematically proven that the sea urchin embryo underwent exactly 12 rounds of synchronous cell division. We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that preformationism was an early, incorrect theory, assuming life just inflates from a miniature form. Second, epigenesis showed us the truth, that life builds complexity step by step from a single cell. Third, we saw how this rapid cellular construction can be modeled using exponential mathematics. Understanding this historical shift is what unlocked the secrets of modern genetics and embryology. You now have a solid grasp of the historical perspectives of developmental biology.